Welcome everyone, to all the attendees of this webinar. This is a webinar entitled Indigenous Food Sovereignty and Contaminants Perspectives from the US, specifically from Southwest and the Arctic. This webinar is sponsored by the Indigenous Food Knowledges Network, the Superfund Research Program at the University of Arizona, and the Center for Indigenous Environmental Health Research. And my name is Dr. Carletta Chief. I am an associate professor in environmental science, and I am the PI of the Superfund Research Program Community Engagement Core, along with Dr. Stephanie Carroll. Our speakers here today are Dr. Jani C. Ingram, who's an associate professor at Northern Arizona University, and Ennoyaq Sudlovenick, who is a PhD student at the University of Manitoba. At this time, I want to welcome each and every one of you, and I hope you are all doing well, safe, and healthy. I'd like to allow the director of the co-lead of the Indigenous Food Knowledge Network and Research to do the introductions for our speaker, and that will be Mary Beth Jager. IFKN.
All right, English, Dr. Chief. Bonjour, I'm Mary Beth Jager. I am a citizen of the Citizen Potawatomi Nation, Chicana and German descent. I am a research analyst at the Native Nations Institute at the University of Arizona. I am, as Dr. Chief mentioned, a co-lead for the Indigenous Foods Knowledges Network. And welcome, glad to have you all here today. And I also would like to take a moment to just acknowledge the homelands. I'm on the Salish, the Puget Salish, and the Snohomish and Duwamish peoples up here in the Seattle area. I've actually been telecommuting for 8 years now, before it became one of the big things to do. So I want to just take a moment so that we all, you don't have to do it out loud, but just to acknowledge where we all are, the people's lands that we all are on in this moment together. And also because today is the first day of fall. And so we all celebrate it, I know, in different ways. I know it's an important season for many of us, Native people. And so just take a moment. All right. I'd like to introduce our speakers. I'm very excited to have them here today with us. So we have, so going first is going to be Inuit. I've been working really hard, getting close, I'm trying really hard, is a PhD student in environment and geography at the University of Manitoba, where she is studying the health of marine mammals through pathology and exposure to pollutants. She has a master's in science and veterinary medicine from the University of Prince Edward Island. Inuit was born in and raised in the Pond Inlet and Iqaluit, Nunavut, and brings together Western science and Inuit knowledge together in her research. And then Jani is a PhD, investigates environmental contaminants with respect to their impact on health. A major part of her research is focused on characterizing uranium and arsenic contamination in water, soil, plants, and livestock. A critical aspect of her research is to foster collaborations with the Native American community and leaders to build trust, obtain access to field samples, and gain insights into their health concerns. She is a member of the Navajo Nation. And so they're both going to present, and then we will do questions at the end. We'll leave about 15 minutes. So please hold your questions. You either can ask them in person or you can drop them in the chat. So take it away, Inuit, please.
Awesome. Qaujimaiatuqngit, Mary Beth, and Dr. Carletta Chief for the intro. I'm just going to share my screen here, and you can let me know if once you see it. Is it visible?
It is visible.
Okay, thank you. Full screen. Okay. Yeah, so I'm Ennoyaq Sudlovenick, but you can call me Inu. I'm currently a PhD student at University of Manitoba, and I'm going to be presenting the work that I did for my master's, which was on ring seals up in Nunavut, northern Canada. And that's why you see both logos there. I'm currently at University of Manitoba, but this work was done at UPEI. And so I'll be talking about mixed methods, Indigenous and Western science in assessing ring seal health. Oh no, it won't let me scroll. Sorry, let me figure this out. Usually you can scroll. Okay, what's going on here? Oh, okay, there we go. So we've moved, we're in the middle of a paradigm shift where research used to be on or about Indigenous peoples. Now we're moving more towards where research is for and with Indigenous peoples, or even by Indigenous peoples, which is awesome. And in a lot of cases, Indigenous and Western methodologies do exist separately. There's a lot of new and up and coming literature out there, some cool papers and books coming out where there are examples of braiding the two knowledges together. I'm sure some of you have heard of like breeding sweetgrass. That's a really good example. And so I was learning how to do this in my master's, and I learned a lot of things. And you'll see some of that during this presentation. I forgot to mention, yeah, so I'm from Nunavut, which is northeast Canada, and it's about 90% of the population are Inuit. So we still do a lot of harvesting marine mammals and like hunting and camping and all that awesome stuff. Just a bit of context on this research. But as someone who did my undergrad in marine biology, I didn't do that much social science stuff, but I learned a lot in my master's, and the difference between methodology, like theory behind stuff, why you're doing it, which populations versus methods, the stuff that you're writing in your papers, like exact how you did it. And I really like to emphasize why a methodology is so important. And in Indigenous methodologies, process is way more important than the actual work itself in a lot of the time. So how you're doing the work is really important. How you're interacting with people is really important. Yes, the results are also important, but process will determine how your work turns out in the end. And I like this quote from Dr. Kolczewski from here at U of M. Northern research involves a twofold responsibility: meeting the demands of academic community and the demands of the Aboriginal or Indigenous communities. And that, I feel like that really nicely captures where we're at right now with Indigenous scholars coming up through academia, and it's kind of a fine balance sometimes. So this project is mixed methods in two senses. And the first sense is that it's Western and Indigenous methodologies, and also it's quantitative and qualitative. So I looked at serology, or blood in the ring seals, and Inuit Qaujimaiatuqngit, or I'll refer to it as IQ because it's really long. But Inuit Qaujimaiatuqngit is Inuit Knoweldges, but it's more than that in that it includes a lot of the values and teachings of Inuit worldview. So it's kind of a huge term that encapsulates a lot of things. But if you're working in Nunavut, people prefer to use this term instead of Indigenous knowledge or Inuit knowledge, just because it includes so many other things and makes it more wholesome. So how this is its methodology perspective, how my data was collected, which population and when and how invasive is it. These three things are the most important things to consider when you're working in the north, particularly in Nunavut and with or throughout northern Canada, Greenland, Alaska. And I will answer those in the next slide. But why I used mixed methods is because you get a better understanding of the research and the research question. It does offset the weaknesses of each method. So if Western science is not very good at placing context with your results, then that's where your IQ can come in and inform the applicability of those results. So they really offset each other's weaknesses. And it's a great approach to complex questions, which in most times, if you're dealing with animals, it's super complex. There's a lot of different factors. And combining numbers and text, it helps bridge the gap between research and practice. So there's a lot of numbers with the serology results, with the blood work, and finding out what that means in the end was really cool. And you'll see that in a couple slides where I have the results. So the question, this is kind of how I came up with my question for my master's. So you have one side, you have Inuit needs, and on the other side, you have scientific curiosity. So coming up with questions, it's really important to listen. I'm from Nunavut, so I'm from Nunavut, and I grew up like hunting all the time and camping. So you got to listen to what people are saying. You got to ask some questions. We have ITK, which is Inuit Dietary Canadami, which represents all the Inuit in Canada. So they're a really great organization. And also look at regional organizations. But you also need to, on the scientific curiosity side, you need to question who does this research benefit. So if there's a question, if you have a question on where are these seals going, then science can find out the finer details, like exactly when and where, and they can make suggestions on methodologies. So my question was, is ring seal safe to eat? And here you can see a picture of a community feast. I think that's in Nunavut, so northern Quebec. So ring seal, like everyone's eating ring seal. I eat a lot of ring seal when I go home. It's harder to get in southern Canada, but that was the question. And so why ring seals? So it is a country food, or you might know as traditional food. People are eating all the time, but it's also a great marine indicator because ring seals are the only seals that stay around when the ocean freezes completely over. So these guys can bore holes into the ice so that they can breathe. So other seals like harp seals will follow the open water. So they leave for the winter when there's when the ice freezes over, which is like 7 months of the year. And as you can see in this picture, this is one hunter. He went out for one day and that's how many seals he caught. So like you get a really nice sample range, and all of it like was given out to his family members. He had a huge family. So yeah, ring seals are a really good marine indicator. And so just a little bit of context as where this was. It was in Iqaluit, which is the capital of Nunavut, and it was spring and fall of 2017 and 2018. That was when I got to go home and sample seals. So the goal was to enhance the knowledge on health of the harvested nutdiq, or ring seal, within Frobisher Bay through context given by IQ. And how did I do that? So my IQ, I did 9 interviews with 9 semi-structured questions on ring sealed observations with super experienced hunters. And I interviewed one of the hunters' wives because she did a lot of like seal processing. And for the serology side, I looked at those different bacteria in the serum. So in the vials there, you can see the serum on the top. So I looked at the harvested population only. There were some questions of people being like, okay, you don't have a representative sample of all the seals. That's true, but we have the ones that people are selectively choosing, and hunters always choose a specific group of seals, just as hunters who are hunting deer, well, they always want the big buck, right? So with us, the Inuit prefer the smaller seals. So it's a very select group of seals that people are choosing. And it, like I said, it was spring and fall. And since these were harvested seals already, we just sampled what they had. So it was like not invasive of live seals. I won't get in this too much just because of time. That's just the methods. So I'll go into the results of these methods, but I did use en vivo, and there were interviews were in Inuit and English, and I did all the interviews and I translated everything into English. So in the end, we did find really cool results. 6 main themes. Like I said, all the hunters prefer young seals every time. If there's a big seal on the ice and you see a big one and a small one next to it, they're always going to go for the little one. They have not noticed a decline in ring seals over time. And they noticed harp seals. So my work was on ring seals, but every time I talk with the hunters, they always made a mention of harp seals and how they're increasing in number a lot, that they're getting to be problematic. And I'll get more into that in a second. We talked about how they inspect seals when they catch them, like which ones they reject, which ones they keep, what illness looks like in seals for them, and which parts they eat and how. So there was a lot of really cool information that we got, but the harp seals was a big concern that the hunters had. So there are a couple quotes here where they're just talking about how every year there's more and more, thousands and hundreds and thousands of these harp seals coming up and taking over the ring seal territory. So if you're going out hunting, you're going to see way more harp seals than ring seals, and that's becoming a concern for Inuit hunters. So like I said, process is more important. So we had communication open the whole time, and that was a bit easier because like I grew up in Iqaluit. I know all the hunters, I know everybody there. So we were talking the whole time. But for the results, I made a YouTube video in Inuktitut with English subtitles to kind of circulate the results. And it ended up being seen in other communities, and they were like, oh, you should do research in our community too, which was awesome. Like everyone, like people are interested, which is great considering that science doesn't typically have a great reputation in Inuit communities. So we disseminated the results, and it was really fun. Hunters were super keen to find out what they were, and we didn't find anything scary with regards to the serology results. Yeah, nothing scary. That's what we told her. I'm still going to keep eating seal, which is the main takeaway. And so the conclusions that I have quickly are methodology is really important. Mixed methods are great because for us, the takeaway we got was that people want more research on harp seals because they're just going to be so many. So that's a new direction for research to go if anybody wants to keep doing seal work. And communication, as we all know, is super key. And yeah, there's benefits and risks to consuming ring seal. Like if you're a pregnant woman, you shouldn't eat the liver of the seal just because of the amount of mercury that's in there. And that's actually more well known in Nunavut, Northern Ontario, or Quebec, rather than Nunavut, which they have different health departments, so they deal with their information differently. But the takeaway is there's a lot of benefits to consuming ring seal. Just be careful if you're pregnant to eat certain parts of the seal. But that's it. Thanks for listening. I tried to keep it in time. I think I did. And thanks to all the sponsors here. We have Nunavut Wildlife Management Board, Garfield Weston, and Canadian Wildlife Health Cooperative. And my mom like sampled all the seals with me. So I put her name there because she did so much work. But yeah, thanks for listening. Okay, I'm gonna stop sharing.
Iqaluit and new. Thank you so much. Dr. Ingram.
Right. Yate. She Jani Ingram. You should be seeing a traditional Navajo food studies first slide, I hope.
Yes.
Thank you. And thank you to Mary Beth and to Bo and to Carletta and Stephanie for this opportunity to share with you some of the science out of my lab. I am a member of the Dene, or the Navajo Nation, on my mom's side. My maternal clan is the Náneesht ézhi, and my paternal clan is Kinlachi, and then my dad is Bilagana, or white man. So it's great to be here. I'm from Northern Arizona University, and I'm gonna talk to you just a small portion of some of the science that we do in my lab. So overall, our lab, I'm an analytical chemist by training, actually from the University of Arizona, go Wildcats. But the work that we do, I consider to be environmental health, where we work with a lot of different folks because it is a very big and interdisciplinary type of approach. And for the most part, we look at contaminants, environmental elemental contaminants, particularly uranium and arsenic, but we've looked at other things as well. And we do a lot of field work looking at water and soil, plants and crops, and then I'm going to focus on livestock today, but all looking at the impact on the Navajo people. So this, if you're not familiar, is a map of the Navajo Nation. It's located in the states of Utah, Arizona, and New Mexico. The sort of smaller lines inside of the nation are chapters or communities. And then, as you can see, the Hopi Nation is also within the boundaries of the Navajo Nation. What you're looking at here is a map made by my student, or my former student, now Dr. Tommy Rock, that tries to show you where some of the uranium mining took place back in the middle 1900s. Those triangles are the old uranium mine features. The circles with the dots are known contaminated wells, which we've done a lot of water work as well. And then specifically today, I'm going to talk about some data that we've collected in the western part of Navajo Nation, which would be in the communities of Cameron and Loop. And then a more recent study that we've been working on in the Cove area. So just a little background, uranium mining, like I said, out on the Colorado Plateau and specifically on Navajo, took place in the mid-1900s. There was quite a few mined attempts anyway, over 1,000, and right now there's like 532, I think, actual old mine sites on the Navajo Nation. There was different approaches to the mining. Some was underground, which is shown in that picture, but also there was open pit mining, particularly in the western side of Navajo. So as a result of all this mining, there wasn't a lot of remediation, so left behind a lot of waste. And this has been an ongoing concern to the Navajo people. Why do we care about this? Well, these contaminants, particularly, as I said, uranium and arsenic, are part of an exposure to the people that live on the nation. I'm going to focus today on ingestion, or in terms of exposure through food, but we've also looked at water, not done a lot of work with dust. The other thing is that there's been studies looking at acute exposure, so really intense exposure at one time, but really where people I think are more apt to be exposed is through a long-term chronic exposure. So in order to do this work, we definitely are looking to get partnerships with the Navajo Nation. And so on Navajo, you get what's called a resolution, which essentially is an approval for your research. And this is something that you go to either the community, or in this case, this picture is my former student, Lindsay Jones, presenting at Western Agency back in 2018. And really essentially presenting your project, and the entire community gets to vote yay or nay. Okay, so I think it's a really important part of doing research, because there's not only just getting the approval, but really those interactions and the dissemination over time. I guess I can turn my video on again. All right, so the big goals of my lab is really to look at these environmental contaminants and with context on Navajo. But we also are very interested in trying to look at this multidisciplinary approach. So like I said, we work with all kinds of different folks from different backgrounds, including tribal leaders and community folks. We also are, one of my dearest, most passionate things in life is working with students, and in particular Native American students, and just being a part of their development as scientists. So our collaborators are many. Like I said, this is sort of a multidisciplinary approach. Some of the most important collaborators really are my students and their families. It has opened our doors to looking at different issues in different communities on Navajo. And we work with the community leaders as well as tribal leaders, some of the government entities, EPA, natural resources. You'll see that I have some work that is human research. So we have approvals through the Navajo Institutional Review Board. Very important to know what other people are doing out on Navajo. Some of you are on this call, so yay. And then, like I said, lots of different scientists that we work with. So I'm going to focus on two different projects within my lab. And the first one is looking at uranium in the traditional Navajo food mutton. So what you're looking at is a piece of fry bread and mutton stew. So if you're not familiar with the Navajo traditions, the sheep are extremely important to the Navajo. They were introduced, as it says, in the 1600s, and the Navajos really became more ranchers, and you can see a picture of folks raising sheep there. It really is more than just an animal. There is definitely a lot of cultural significance. Having some mutton is not just like having a burger. So the issue of contamination within the sheep that are being raised on Navajo is very important. And part of the reason we got into this research was that we had community members asking this question to us over and over. And finally, we had the opportunity to sort of delve into this with some EPA funding, and then more recently with the National Institute of Environmental Health Sciences out of NIH. So our focus is really to quantify the uranium in sheep, and not just in the sheep, but in different pieces and parts of the sheep, because we are interested in trying to help people know maybe a specific organ or tissue has higher levels than another, so maybe to stay away from those. We've also worked with the Swinomish tribe and looking at using their Indigenous health indicator to really look at the health impact. We're still working through that. And then, you know, really what are those uranium exposures? So what you're seeing to the right side of the slide is a map. Again, Navajo Nation shown here. Flagstaff, where I'm located, is about right here. So we collected sheep from Cameron, which is the black dots, from Loop, which is the red dots. And then we looked at this site far off of the Navajo Nation, and you'll see in a few moments why that was, in Eager. And then more recently, we've been up here doing sampling in the Cove area. And the yellow dots are trying to indicate the abandoned mine sites. So you can see there was a lot of mining in the Cameron area, seemingly none in the Loop area, and then a lot in the Cove area. Cove really was underground mining, and then Cameron really was the open pit mining. So in order to do this work, we definitely worked a lot with our community folks. And so during our sample collection, really we called it a butchering party. So we would go and we have statistically determined that 5 sheep from a flock is fairly representative. And so it took an army of students really to go out and to collect the list of tissues and organs that are shown there. And so actually, I think someone on the line, this is Marcia Bitsui right here, one of my former students. But this was something that we did with all of the collection. Was it really was a partnership with the community folks. So I'm just going to cut to the chase, and that is looking at some of our results. So these are results from comparing sheep collected in Cameron, which again, there was the open pit mining, to Loop, which is also in Western Navajo, but there was no mining there. And then to this off-reservation site in Eager. This is an average of 5 sheep, and we're looking at the kidneys. And so what you see is actually higher levels in both Cameron and Loop, and then the lower levels in Eager. So initially we were going to use Loop as our control site, but when we got the results back for pretty much all the different tissues and organs that we looked at, the levels in Cameron and Loop were very similar. Now we don't know what's background or normal because those numbers are just not available. And so then we have the question, well, is this just background levels? And so that really isn't an effect, or is actually Loops seeing elevated levels even though there was no mining in that area? So that's what motivated us to get 5 sheep in the Eager area. And as you can see, it is factors of a couple to 3 or 4 times lower. And so it does seem that there is some additional, or there's some uranium in these Loop sheep. Now, it turned out that we found out there's a lot of sheep trading, and we didn't know exactly what areas the animals were grazing in Loop. And so then the follow-on study actually was to look at sheep in Cove. That study is ongoing right now, but that study is a partnership with the University of New Mexico, with Montana State University Billings, and with Dinek College, with Dinek College being the lead. And that project, we're not only collecting livestock, we're also collecting environmental samples. But the important thing is that we are actually collaring animals in order to do a GPS tracking of where they are grazing, so that we can pull environmental samples exactly in those areas. So that, like I said, that study right now is ongoing. We hope to be a little bit further along, but COVID sort of put a kibosh on some of our sample collection last spring. But what you're looking at here is just comparing the Cove information that we have to date, which is 1 goat and a couple sheep. This is the backbone meat on the animals. Then we compare it to Eager, Cameron, and Loop. And we do see actually quite a bit lower level in Cove. I'm pretty careful, cautious in trying to show that now, because like I said, we need to do a lot more work to see if that's true. This could potentially be because there was underground mining here, and in particular Cameron, it was open pit mining, so it's a little bit more exposed, but we're still trying to understand that. So one of the other things that we did, part of the study, talking about a scientist trying to do social science, is we wanted to know how much people were actually eating of mutton. And so again, my former student, now Dr. Tommy Rock, as part of his dissertation work, he did a survey of folks in Cameron looking at their consumption behaviors. So we had to have a lot of approvals in order to do this human research. But what we found was he surveyed 72 people. The demographics are shown here. We had a pretty good representation from young to old, with the most folks being between 40 and 70 years of age. The folks were not all exactly in Cameron, because what Tommy did was he went to events that were held in Cameron, and sometimes there would be other folks present at those events. But overall, I think he got a pretty good feel for the mutton consumption behaviors there in Cameron. And what we saw was that people don't eat as much mutton as they used to. It's mostly at family gatherings, so maybe monthly or a little bit more often. Definitely older folks eat more mutton than younger folks. But people were stated that the mutton was very important in their traditions, and they really wanted to be able to continue being able to eat mutton. So what we have done recently is actually did this same survey in the Cove area. Cove is about the same size of community as Cameron, except it's on the east and in the northeast. So we're going to be comparing those results and really trying to get a better feel for mutton consumption across the Navajo Nation. So the other study I wanted to just briefly talk about is one that was done by a student named Daniel Begay, looking at calcium in blue corn mush. So that's a picture of blue corn mush there. Juniper ash is added to a lot of traditional Navajo foods. It seems to be a potential source for calcium for folks. There was a previous study looking at fractures in elderly Navajo, which was much lower than in Caucasian women. And so it was hypothesized that because a lot of elderly Navajos eat blue corn mush almost on a daily basis, this could be a very good source of calcium. So we wanted to investigate if the juniper ash was what was adding calcium to the diet. So his work was to quantify calcium. He bought juniper ash at flea markets, but we also collected juniper, and he ashed them both in a couple different ways, using a furnace, and then also he learned how to do traditional ashing. So this is just pictures of putting the ash. This is after it came out of the furnace. It was nice and white, and then he actually learned from a couple grandmas how to burn the ash burn the juniper in order to get the ash. So these were some of the sites he collected his juniper. Part of thing we were interested in were certain areas higher in calcium than others. And so what we found is shown here is these are all these different samples, and essentially there is some variation, but for the most part it's 250 milligrams per gram plus or minus about 50. You can see that his laboratory ash seems to have higher concentrations than the traditional. However, if we looking at his juniper ash that he got from the flea markets, it's also fairly high, 250 to 300. And so the good news there is that we think that that you really don't need to go to a certain area of the Navajo Nation to collect your juniper to get good calcium concentration in your ash. The other thing is Daniel himself thought that he probably didn't do the traditional ashing as well as probably one of the grandmas. So that might be why his laboratory ashing was higher. What we want to do next, I have another student who's going to actually look at calcium in a few different sources of blue corn that people use. Since most of the blue corn mush is blue corn, it would be useful to know how much calcium is coming from the blue corn. And we are in the midst of collecting those results in order to publish these results. And then the last thing I'm going to just refer to very quickly is that super important is dissemination, and then also some of the work we've done in policy. So these are pictures of folks from Cameron who came to visit our lab. This is my former student Tommy Rock, who invited them, and we gave them a lab tour and had a discussion. This is my other student, current doctoral student Andy Lister, in with the instruments, kind of trying to explain how we go about doing our science. Then Tommy also got funding in order to actually put on a series of conferences with respect to uranium issues out on Navajo. And he held 4 different meetings. As you can see here, this is a picture of some of the folks in my group in Cameron with State Representative Peshlakai during one of those meetings. We also have been called upon by different political folks in order to either provide our insights during, in this case, it was Representative Tom O'Halloran, and he was having a town hall in Cameron. And so shown down here at the table is one of my former students, Jonathan Credo, providing some background information on our work. So I'm going to leave it there. We have, like I said, lots of help from different folks, and the funding agencies, EPA and NIEHS, have been very generous. We also want to thank our collaborators in Cameron Cove and Loop. We've done some work, particularly Tommy, with working with the Dene Hatatli Association, which is the Medicine Man Association, in terms of thinking about how to do this work culturally correct. And so these are a lot of my non-biological children that I've had over time, but I wanted to leave you with this last slide, which shows different books if you're interested in learning more about the uranium issues out on Navajo. So with that, I thank you, and hopefully we have enough time for some questions. Mary Beth, you're muted.
Thank you for telling me that. So I want to say thank you to both Jani and Inu. So either use your reactions or a round of applause for wherever you are. So thank you so much for your presentations. I think we can do questions if someone wants to ask one. If you could either put it into the chat, or you can also, there's a raise your hand function. It might, because we might have just enough people that it might be a little too unruly to have just people start speaking. So there's a raise your hand if you go over to the participants too, that we can look at. So either put it into the chat or raise your hand. And
let me ask one question, because if we look at uranium, I mean, we're going to find much more uranium in the bone. Have you looked into soups perhaps that they use with mutant bone?
Yes, so we've looked at the bone itself. Bone is really hard to do the analysis. There's just so much calcium, it's a problem, but we've done some of that. So you're right, bone is where you have collection. And then we have looked at leaching essentially in stews, because that's a very important part of the Navajo diet. And we compared it actually to looking at the same sort of rib meat type with the bone present, frying it, boiling it, which would be the soup, grilling it, and roasting it. And it does seem like you do see more in the soup. We're still trying to parse that out, but it does seem like the others were sort of at the same levels, with the soup being at a higher level. So we want to make sure that we didn't introduce anything with the water that we used and other things, but that does seem to be perhaps a concern, which is not good news, just because that's a very typical way to feed a lot of people out on Navajo.
Dr. Chief.
Thank you for that excellent presentation to both Ennoyaq and Jani. My question was to Jani for the analysis of the juniper ash. How did you analyze that? Did you separate the berries from the actual stem and then just look at the leaves itself, or was it all together? And would you, if you were to separate it, where would you think would be the highest amount of calcium?
So let me share really quick. I can do this. Put it over here. So I didn't put that one up, but if you see looking here, this was just the juniper berries. And then the red is just the branch. And so the branches have a bit more, but it's not a lot more. And then most of the ashing that he did though was all together, because he thought that the grandma that he was working with, that's how they typically do it. So you do see a little bit more in the branch than the berries itself, but at the same time, it's not, you know, it's not a lot. Oh, did I not share that? Sorry. Can you see it now?
It's on there, yeah.
Okay, all right. Yeah, so he actually did that work as well.
So we have a question. Thank you both for presentations. Are the CA levels you see ash samples and the amounts of foods consumed high enough without inputs from blue corn to support bone health in the elders?
So that's why we want to look at the blue corn to see if we also have calcium, and then to kind of start thinking about equivalents of, you know, what the calcium daily requirement is. So, but my understanding is, I mean, because it's so easy to both chew and digest, that, you know, many senior citizen homes have blue corn mush every day for their folks. So it's a very prevalent food. And so, but we need to understand where, if there's very much calcium in the blue corn.
Thanks. So Nora Johnson has a question for Innu. What questions and concerns have Inuit raised about contaminants in country food? How have their questions influenced your next areas for research in your doctoral program?
Thanks for the question, Nora. Yeah, so there were concerns about mercury mostly, and that's kind of a recurring contaminant that we're hearing about even right now in my doctoral program, where I'm looking at beluga. And so mercury seems to be the most, I guess that's what you are worried about right now, is mercury. And we looked at mercury in mycelials, and we looked at total mercury and also methylmercury. And what we found is what we were expecting, and that muscle doesn't have that much mercury compared to, say, the liver, but people are eating the liver, and they're also eating the meat. But we did not look at blubber, and people eat a lot of seal blubber, so much so that they eat it with other dishes. So if I were to continue with ring seals, I would look at mercury in blubber. So that kind of influenced future directions of research. Yeah.
So we have some comments. Carol said, interesting about the berries. The grandmas I know do not include the berries for the most part. It's the juniper and cedar that has some red rust color on it that they prefer. Fort Defiance and Canyon Deschutes areas. So the family community differences even with ash. And then Dr. Chief commented that at the hospital, they don't serve blue corn mush with ash, that she asked when they served it in her room. And then Susan had a question for Jani. Is it known what the path of contamination of the sheep is, water, or what the sheep eat?
So that's a good question. We have looked at unregulated or livestock wells out on Navajo. So it just depends on the well that the sheep are drawing their water from. And then definitely we've looked at plants. And this is still ongoing, but if the soils are higher in the uranium, then the plants tend to be higher. At least on the Cameron side, there's a grass that we looked at, and unfortunately the uranium accumulated more in the shoots, so in the top, than they did in the roots, which a lot of times the metals will accumulate in the roots, and so unless the animal is really pulling all the way down to the roots, it's not too bad. So, but I think it's a combination. I don't think it's a simple answer.
Thank you. And then Hannah says, thank you for the excellent presentations. Jani, did you analyze the juniper for other nutrients in addition to calcium or any potential toxins, contaminants like PAHs from the process making the ash?
No, we just looked at calcium, but it would be interesting. We were going to try and work with a biologist to look at some of the proteins in corn, because I guess there's a possibility that even the corn would play a role in terms of bioavailability, but we haven't connected on that study. So hopefully we can learn more about it.
Nor has another question. It's for both Jani and Innu. If there is a time for more than one question, Innu in the Arctic, I know there's a lot of concern with long-range transport of contaminants. Are there local sources of contaminants that are also a concern? And then Jani, are there any concerns about long-range transport in Navajo or just locally sourced contaminants?
Do you want me to go first?
Sure.
Okay. Local sources, yeah, so we don't have very good sewage systems. So they do leak quite a bit into the ocean, and a lot of people will be fishing or harvesting shellfish close to the towns. So that is, I think, the biggest local source of pollutants. But there are, I know in North Baffin Island, we have an iron mine up there, and there's a lot of shipping. So the main concern there is ship noise for local disturbance. Yeah.
So on Navajo, besides the legacy uranium mining, there's current coal mining. So there's issues with respect to that, although we have done a little bit of work looking for mercury in different types of samples and haven't seen high levels or levels above background. The other thing is there's also oil and gas. That's particularly in the northern part of Navajo. And so there is definitely concerns about organics there. My group doesn't do organics, so we haven't done a whole lot of sampling there, but there's certainly other contributions to some of the contaminants that folks are concerned about.
Great, thank you. Carol Heck for a question. For Navajo, arsenic is also a concern. Is that on the horizon for your research?
Actually, we do a lot of arsenic, and quite honestly, particularly in these unregulated livestock wells, it's probably a bigger concern than uranium. We've seen it in, as in the wells that we've sampled, which are more on the western side of Navajo, is 30–40% of the wells have elevated levels of arsenic. So yeah, that is definitely something that my lab looks at a lot.
And so, Dr. Chief, to follow up with Carol, arsenic is linked to diabetes. Did your survey percentage people add sugar to their blue corn mush?
So we haven't done any surveying on the eating of blue corn mush, which would probably be a good thing. Mostly we've done the mutton consumption survey. So that's something we should probably look at. I know for myself, I add sugar, so it's pretty tasty that way, but I know that just adds to the calorie count if nothing else.
Are there more questions? I had a question for both of you to answer about working with the tribal governments or the First Nation governments. Is this something that they're part of the project, or did you present? I know you talked about presenting like the video in new. Is there something more or something that they're interested in working with and collaborating you with?
Yeah, so for Nunavut, we have to get approval from Nunavut Research Institute, and you also have to have local approval through the hunters and trappers organizations. So in most cases now, research won't get funding if you do not have the hunters and trappers support. So there's like the local organization, and then there's the regional organization, because Nunavut's split into 3 regions, and it's different for like Northwest Territories has its own set of organizations too. But yeah, there's quite a few.
Yeah, and on our side, definitely, especially the mutton, the livestock work has been a huge push from the community, asking that question if there's uranium in our mutton. But that new Cove project actually it's funded through EPA, but it's actually from a settlement from a uranium company to the Navajo Nation. And the community was asked, you know, what do you want this funding to go toward? And they were very interested in uranium levels in Cove in their livestock. So that's why we also are looking at goats, and we are looking at cattle. Cattle are very different. Cattle are more of a cash crop, so to speak, for those folks. So we're also going to be collaring cattle and seeing where they graze and see if they're being exposed. So yeah, but in order to do particularly the livestock work, if the community is not engaged and working with you, I don't see how you could accomplish it. So it's very important.
Great, thank you both. Any more questions? So it doesn't look like there's any more questions. So thank you once again to both of you for presenting and for sharing your work and taking the time today. Very much appreciated. So another round of applause. Yay. Thank you so much. I do really appreciate it and learned a lot. So, and thank you for all of you who attended. We do plan on putting the recording up on the IFKN website, which is IFKN.org, and I think we're also looking at putting it, I think either the Superfund or other site will have it too, so the center. So look for it there. All right. Thank you. So yes, Innu and Jani, I'm gonna eventually get your name correct. I'm so sorry, I stumbled over the words in your bio. I felt really bad too. Yes, so thank you very much again for both of you for presenting. It was really fascinating to hear your work. And because of the work they've been doing up in Swinomish, the health indicators is something that's come across my desk. And I've been really curious about, because Swinomish has been really a leader in like climate change and stuff like that in their climate adaptation plan. So that's been.
Right. Yeah, no, it's been great to see the overlap and the interest among tribes. So yeah, well, thank you for the opportunity, and it was great to hear your work,
Yeah, thanks for having me. I was like, oh gosh, so many people, but this is fun, and it was super cool to hear about your work, Jani.
So yeah, thank you. And so I guess it's a question. Is a harp seal something that you would normally, is that also considered a country food, or is that just sort of it?
I think people only harvest those for for dog food, like if you have a dog team, but people don't typically like to eat them. They're huge. They're too big.
Are they, I don't know if you have them. So we have harbor seals down here. Are they like harbor seals? What? Or harbor seals are small?
Yeah.
Okay. So they're way bigger. Okay. I'm getting used to living on the coast. I wasn't on this coast, or in this region, so on the coast side. But I grew up in like the center of Oregon. So that's I know a little bit about marine life, but not tons. And my tribe's Great Lakes area, and then we were moved down into Oklahoma, so it's lake life. So very cool. All right. Well, it's 1 o'clock, so I'll let you, please feel free to leave.
All right. Well, thank you.
Thank you. Bye.
Bye.